content and all these things. Okay, so what have to be the characteristics of E. coli, characteristics of coliform? So normally what are the characteristics? Characteristics are enterobacteria. For example, you can look at the coliform genera or enterobacteria. What have to be the characteristics? Normally if we talk about the coliform genera, in the coliform genera we found different part. Let me change the color. So we can look at in coliform genera the Enterobacter, Klebsiella, Escherichia, and all this will be the among the coliform genera, as we've talked about. Escherichia is a very common one. We've talked about Klebsiella, we talked about Enterobacter. So what are the characteristics of this coliform? So characters are, they are facultative anaerobe. That means they live in without the presence of oxygen, but if the oxygen present, the oxygen cannot act as poisons for that. They can also live in presence of oxygen, but without oxygen they can live really longer. It's optimum for them to live without oxygen. They are gram negative in nature, so if we stain them with gram stain, they are gram negative on those cases. That means in their bacterial cell membrane layer they have 3 membrane layers. They have a peptidoglycan layer which is really thin, and they have an inner layer and they have an outer layer which is LPS layer, which can act as endotoxin too. And they are non-spore forming. Normally these enterobacteria are non-spore forming bacteria, so they cannot form spores, but there are some type of spore formers that I have talked about, this perfringens type, which are spore formers. But in this case the coliforms are non-spore formers. They are normally rod shaped. For example, as we know the E. coli structure like this, they are rod shaped. They can ferment lactose. That's a very important property, the biochemical property of them. They can ferment lactose, and as they can ferment lactose, this feature of them, we use this feature of them to utilize them against some different biochemical tests to figure out whether the presence of the coliform in a water is there or not. So we use lactose medium there, and we used to add different water samples on them. If we have coliforms, a significant amount of coliform there, that they can ferment the lactose, and as a fermentation of lactose done, they will produce carbon dioxide, and when they produce carbon dioxide, that carbon dioxide can be seen. So by determining the production of carbon dioxide and the amount of carbon dioxide that they produces, we can determine whether the coliform is present in the water sample or not. Okay, and they can also produce gas, as I have told before. We can produce gas within 48 hours at 35 degrees Celsius temperature. We can observe this at 48 hours at 37 degrees Celsius temperature too. So these are some of the characteristics, and the last 2 characteristics are used for biochemical tests to determine the presence of coliform in water sample. Okay, let's move on to next slide. Why it's not going? Okay, yeah. So finally what we have, this indicator organisms. So we have a summary. What the summary is? Summary is for the general coliforms, which are the indicates the water in contact with plants and animal life, which is called the general coliform. So what do you mean by the coliforms? Coliforms are the some sets of bacteria which indicates the presence of pathogens, but the coliform normally means that the some types of microorganisms that have in direct contact with plant or animal life. Now what are the fecal coliforms? Which is the contact with mammal or bird feces in water. And what are called the enterococcus bacteria? They are type of different fecal streptococcus. They can found in feces also. So here are the examples of this. So this is a streptococcus example. Here is the example of Escherichia coli type of bacteria, which has the normal fecal coliforms, and we have a huge amount of different coliforms going on. Okay, so these coliform bacteria is generally not what making people sick. So we have other organisms like pathogens which cause diseases like Vibrio cholerae, like Salmonella typhi. So we can just have prove if we have these coliforms in the water that there must be different pathogens. Okay, so what are the typical water quality standard for a purpose? We have talked about that. We have drinking water, public safety water, recreational water, fish and wildlife. We have talked about this in before slide, so it's a double duplication slide, so just skip the slide. So what are the tests for pathogens? There are several different tests for presence of pathogens. Normally there are simple direct tests, but I have told you that direct tests are really time consuming and they're expensive, so we cannot do direct tests because we need to give the report under 24 or 48 hours of the contamination water testing. So we need to go through simple different kinds of tests that will tell us what is the chemical composition of the water, what is the presence of coliforms in the water. So we are using the different chemical tests. So normally in this defect stages, normally in this kind of stages, we have involved selective cultivation of large numbers. So in this case we need to follow cultivation of all these things in large numbers in laboratory conditions. We need to culture those in laboratory conditions. They are expensive, that's why we do not use them normally. So molecular steps are there which require testing of each pathogen, testing of each simple pathogen, which is really expensive. You go to PCR technology, those gel electrophoresis technology, and all those technologies really a hard time to figure them out, and really expensive too. So that's why I do not use this, because they also need expertise to handle with this. So that's why we do not use them. What we are using, and nowadays we are using the chemical techniques to do them. So finally, first thing, if we want to remove all those microorganisms from water content, we can use filters. So we can filter those microorganisms, and when you filter those microorganisms, then you can find the microorganisms is entered in the filters. So we have these filters. They can be nylon filter, they can be diatomaceous earth filters. So all these filters, we have a funnel here. Let's talk about the funnel. Throughout the funnel we add water. When we are adding water, we are adding contaminated water, then the water comes out easily, and the bacteria are trapped in this filter. So we can find this filter, and we can check this. We can analyze this filter. After analysis, we can derive results, whether the water is contaminated with microorganisms, with contaminated coliforms or not. Normally the diameter of the filter, or of a membrane filter, is 0.45 micrometer. Okay. So normally we can place this filter into selective media to grow, incubate specially coliforms. So suppose we are doing this experiment like which is drawn in this picture. After doing it, we take the filter, we just place them into selective media. What is a selective media means? Selective media means some type of media which will support the growth of a selective type of microorganisms. In this case, we are checking for coliforms. We're checking for coliforms. If we add some medium which supports the growth of only coliforms but growth of no other microorganisms, then the coliforms will grow in this in the agar plate really rapidly, and from them we can determine whether the water sample we have examined contain any bacterial coliform content or pathogenic content or not. We do not select this media to grow pathogens, because this is hard and this is really dangerous to work with pathogens in laboratory situations. It needs expertise, it needs very tough technology, it needs expertise too. And we after incubate them in selective media, after placing this paper in selective media, we need to incubate that for 37 degrees Celsius temperature for almost 24 hours. And if we want to talk about the fecal coliforms, we will incubate them in slightly higher temperature for 45 degrees Celsius temperature for 24 hours. After incubating them, we can check for the presence of coliforms in the farm. So we can find colonies. If we found colonies, we have to count those colonies. So we have a relatively lower count of colonies, and the counting of those colonies, we can determine the amount of coliform that present in every 100 ml of water. And if we have no coliforms, then you can say the water is safe and we can drink that water. If we have higher than 200 and 2000 and all these things, we can say almost like 200 coliforms per 100 ml. We can say that the water is what you can say, it's a recreational water. For example, if we have more than 5000 count, we can say this is a non-drinking water. This water is the habitat of fish and wildlife. And if we have a lower, like 14 or 15 coliform, we can say shellfish can grow on them. So we can conclude after this kind of experimental setup. Okay, this is a direct type of experiment with filter method, so it's a good reliable method to do that. But some of the microorganisms, if they passes through this membrane filter, we can determine. So this check is actually possible for different coliforms, but actually there can be viruses, there can be smaller microorganisms that can passes through this diameter of filter range. And what we'll do in those cases, we use another set of different possible possible type of experiment we call the multiple tube fermentation, or NTF, or multiple probable number, or MPN method, to detect the amount of coliforms, the density of the coliforms in that, and the density of the coliform is correlated with the density of pathogens. So if water have a content of higher coliforms, it is probably the water is contained water is having higher content of pathogens. So what we are doing a serial dilution to extinction. What is a serial dilution? In the serial dilution technique, we have several test tubes. In the several test tubes, what we are doing, we are actually taking for example 9 ml of broth, or liquid or nutrient broth, for example. 9 ml of water, distilled water, 9 ml of sterile water, for example, 9 ml of sterile water. In all those tubes we have 9 ml of sterile water. What we are doing in this case, we are adding 1 ml of water to be tested there. The water contains the coliform. Then after making it 10 ml, we take 1 ml from here and add it into second aid, and then take 1 ml of here, add the next one, 1 ml next one. So this is called the serial dilution. Why it is called the serial dilution? Because we are diluting the amount of coliform. So if we have a coliform of 10 to the power 5 here, then after the first dilution we will have 10 to the power 4, in the second dilution we have 10 to the power 3, we have 10 to the power 2, and 10. So we have the coliform counts is diluting like that. Okay, so that's why we call it the serial dilution. And what is the purpose of doing the serial dilution? Because after we plate the dilution of this into an agar plate, and we finally find some colonies, by determining the amount of colonies, the number of colonies, or sorry, not amount, the number of colonies, we can determine the actual density of coliforms. So we can find the density of coliforms by doing the serial dilution test. Same as the normal process, we can incubate the 35 to 37 degrees Celsius temperature for total coliforms, and 44.5 degrees Celsius temperature for fecal coliforms to grow these colonies. And when you find these colonies, we take the number of these colonies, and finally we can determine the amount of bacteria present in the actual sample what we have taken. Okay. So why does it has another implication, that is if we take this actual sample, if we plate it directly, then you can find probably large amount of colonies, because one simple bacteria can give rise to a colony. So that's why we talk about this colony forming units equals to one type of bacteria. So that's why if we have a lots of bacterial or pathogenic coliform contained in this test water, then they will spoil this plate, because they will grow rapidly. We can count the colonies. So we need something, some range of countable numbers of colonies. That's why we are creating a range. We are actually excluding the very dilute parts, and we are excluding the very denser parts. What you're taking in the middle parts, we are taking samples from the middle parts, middle dilutions, and we can find the some colonies which is really countable, and by counting those colonies we can determine the actual density of the coliforms, and by determining the actual density of coliforms, we can determine the actual density of pathogen in that water content. And we also use different types of coliform tests. Now it's a ready to available test, like this Mendo test. In this case we use basic fuchsin and sodium sulfite, which forms a metallic, which finds a green metallic sheen on the colonies, able to ferment lactose to acetaldehyde. Okay, so we have the medium. In this medium, what we contain in this medium, the medium contained what we have, the lactose fermenting bacteria. So medium contain the lactose in it, and it also have the basic fuchsin and sodium sulfite. So what they produce, if somebody ferment lactose, they'll produce acetaldehyde. If acetaldehyde is produced, then the acetaldehyde can react with basic fuchsin and sodium sulfite to turn the color of this is a metallic sheen, green metallic sheen, and that can be formed in these cases. Okay, so it's a basic fuchsin contained medium, that's why the medium is red in color. So after doing the experiment, we add water. We have water. If the water contains coliform, then it will ferment lactose, because as we know, coliform can ferment lactose and it will produce gases and acetaldehyde. So when it is produce acetaldehyde, the acetaldehyde will react with this sodium sulfite and turn the color green metallic sheen. Whenever we find green metallic sheen with this Mendo experiment or endo agar, we call it the endo agar. So that's why we call it the endo agar. So after having having mecon is endo agar, the color change, the green metallic sheen coloration in the agar, we can find, we can say that the water sample contains some of the coliforms. It may be total coliform, it may be fecal coliform, but we can say it contains some coliforms. And it also can have this take media, which is non-target colonies turn purple color when baked down urea to rise the pH. Same again in this case. So we have normal colonies which is white in color, and we have some colonies which are really which are purple in color. So some of them are purple in color, which we are looking at this case. So why they are purple in color? Originally they are yellow or orange like that. Some of them are purple. Why they turn their color into purple? Because they react with the breaking down portion of urea, and the pH is rising up. When the pH rises up, that color of the media, take media, will become the purple of the colony becomes purple. So we can determine whether the fecal coliform present or not. So by this earlier experiment we can determine whether there are coliform present or not. We cannot distinguish between a fecal coliform and total coliform, but in the second experiment where we use the take media, we can determine whether the E. coli present or not. If E. coli present, the E. coli we know as is a fecal coliform, then you can say the water is contaminated with the fecal coliform, and it is not very safe to drink. And we further go down different experiments through which we can determine the density of the coliform, and then we can actually conclude our results. Okay, now let's talk about different enzymatic reactions to determine and distinguish between different forms of coliforms. If we look at these, so via the previous steps of reactions we can determine, or the different presumptive tests are there to determine whether we found any coliform in the water sample or not. Via those reactions what we have talked about, that we use Durham's tubes. So we have the test tube, we add those liquid broth, and this liquid broth we add Durham's tube, which is a simple small tube, and we put the Durham's tube such as they can be put in this opposite direction or on the opposite orientation. After putting it there, as we know that this materials, we can ferment this as lactic acid portion, and it will produce carbon dioxide. If there is coliform bacteria present in this water, they can ferment this solution, this medium, and can produce carbon dioxide. So if they produce carbon dioxide, the carbon dioxide is going to get upward, it is going to store under the Durham's tube. So after that we can find any result in Durham's tube. If you look at the Durham's tube, then what we can find, you can find somewhat air, which is carbon dioxide, is stored in this Durham's tube. In many cases small, in many cases larger, or in some Durham's tube it may not be stored. So from looking at the production of carbon dioxide, we can say that the water is contaminated with fecal coliforms or coliforms, not actually fecal coliforms. It actually helps us to determine whether we have coliforms or not, but it do not helps us to determine whether we have a fecal coliform or enteric bacteria genera. So after knowing this, we can determine different, we can have another chemical steps which is going to tell us the presence of the total coliform or in the presence of fecal coliforms. They are chemical tests. So these are first normal steps or presumptive tests that's going to easily tell us whether we have coliform or not, using by this air carbon dioxide production tests. But without these tests, if we want to distinguish between fecal coliform and normal coliform, because we have a coliform is a large genera, and among them we have a total coliform, and among them we have a fecal coliform count, which is an E. coli, and in this total coliform we have Klebsiella, we have Enterococcus, Enterobacter, and all these types of bacteria. So how can we determine between those genera of different coliforms? We need to go through another different sets of reactions. So in these future slides I am going to discuss about the process of determining whether we have a fecal coliform or a normal coliform. So we have first, let's talk about the enzyme substrate or chromogenic substrate reactions to distinguish that, because as we know, if we have coliforms, the coliform has the enzyme beta D-galactosidase. So it has the beta galactosidase because they need to break down lactose. So in this case what we are using lactose medium. So not only they use lactose medium to produce to ferment that lactose medium and produce carbon dioxide, that's going to tell us that this water sample contains this this coliforms. But also we have another type of identification purpose. Here we have beta galactosidase, which is secreted by the cells, which is secreted by this coliform cells, which hydrolyzes an enzyme which we add which hydrolyzes not enzyme, a chemical substrate, ONPG, or its full form of it is orthonitrophenol beta D galactopyronoside. So it's a really mouthful of name, so I recommend you to remember this name, ONPG. So when the ONPG present there and beta galactosidase ferment the lactose, it breaks down or hydrolyzes the lactose, it will produce allolactose and glucose and galactose, and it will react with those glucose and galactose, and finally it will produce yellow colored substrate. So whenever we have a yellow colored yellow coloration in our broth, then you can say that there is total coliform present in it. So with an enzymatic assay like that. The first assay I have talked about here is a gas production assay. The second assay is enzymatic assay. So this enzymatic assay is not only help us to determine the total coliform, but it can also help us to determine whether fecal coliform like E. coli can be present in water or not. For doing this purpose, as we know, in case of E. coli, it will produce beta glucuronidase along with beta D galactosidase. That's why they also produce this beta glucuronidase. So normally E. coli or fecal coliforms have the only ability to produce beta glucuronidase, but other coliforms except fecal coliforms do not secrete this enzyme. That's why if we have this beta glucuronidase secreted from the E. coli, or you have E. coli in the water solution, water sample, then which this beta glucuronidase will hydrolyzes another important chemical which we add in those medium, which is called MUG. The full name of it is really complicated, so